To the world today. So, hi to everyone, and we'll get right to it. So, Professor Miller, up first.
Thanks, Ryan. All right. Thanks, everybody, for coming. I'm just going to tell you real quickly about a project I've been working on with Scott Carter on cancer, phylogenetic inference. So as it develops within each patient, cancer evolves into multiple subpopulations. So mutations are occurring all the time, but occasionally some set of mutations will confer a selective advantage, and then a new subpopulation will arise, and that's what's depicted in this picture here. And these subpopulations are called clones. So here's a cross-sectional image of colon cancer, and the different colors represent different subpopulations that have been identified visually. So this is a very common characteristic of cancer, and we can model it mathematically or probabilistically with a phylogenetic tree in which mutations are occurring as you go along the tree, and then they're passed down to subsequent populations. So like this red mutation is shared by all of them, the green one is passed down. There's a variety of different types of mutations that arise. Point substitutions where a single base is substituted. Small insertions or deletions where one or a few bases is added or removed. Larger copy number alterations where a whole segment is duplicated or deleted. And also translocations where a piece gets moved from one chromosome to another. And so cancer genomes end up being extremely sort of messed up. On the left is a normal one where you have 2 copies of every chromosome, except for the sex chromosomes. And then on the right is a cancer genome where there's several copies sometimes of some chromosomes, there's pieces missing, and you get also these kind of really crazy combined chromosomes. So this is a particularly nasty example here. All right, now if we wanted to use genome sequencing to understand cancer and to help with the treatment of a given patient, then we could take tissue samples and do whole genome or whole exome sequencing. But a challenge is that if you take a tissue sample, then often it'll contain cells from multiple different subpopulations. Or if you take a blood sample with cell-free DNA, it's a similar type of problem. So the challenge is that if each sample has cells with DNA from multiple different populations, then what you see in any given sample is not a clean genome. It's really kind of a convolution of multiple distinct genomes. So when we look at the data, this is focusing on loci that have been identified as classified as mutated, and this is a couple hundred or so of such loci, and showing here in the log counts of mutated versus normal reads at those loci. And so for a given patient, you typically will take multiple tissue samples, or hopefully you will get multiple tissue samples from the same patient. So that's showing the counts of mutated loci. You also get what's called copy ratio data, which quantifies the changes in copy number at different segments in the genome. And so we get these 2 different kind of main sources of information. So the blue and the red here are basically different homologous chromosomes. Sometimes there's amplification, sometimes there's deletions. So the overall goal that we want to solve is to figure out how many different populations are represented in a given data set for a given patient, that is. What are the mutations that are present in the different populations? What's the phylogenetic tree that describes how these different mutations arose in that patient? And the motivation for solving this problem is the science. On the science end, we want to understand how cancer is developed in general, what mutations arise at what point in time. And for medicine, we would like to be able to tailor treatment to the particular individual. So if you can get these samples and say, these are the mutations that occurred early on, these are the ones that occurred later on, these are the different populations, then you could choose a treatment which is particularly effective for that person. So our approach for this is using a Bayesian model for the phylogenetic tree, where the mutations are occurring on the tree, and then for the observed data given that latent structure. The main challenge is, so so you can write down that model. The main challenge is doing computation. So there's some strong dependencies introduced by this tree structure, and it makes the space quite difficult to search. So anyway, so we've developed an approach to deal with this computational challenge, and let me know if you have any questions.
We have time for questions, so.
So, quick question, Jeff. So,
yeah.
in the way you've sketched this, there's sort of an initial mutation, and everything's sort of a founding mutation. All the clones contain that mutation. I've seen some evidence that in prostate cancer, actually, multiple tumors will develop with different founding mutations. So it's sort of like you have multiple independently developing tumors. Do you see that evidence in other cancers?
I think, yes, I think there is evidence for that in other cancers as well. Yeah, and so you need to have a model which allows for sort of multiple trees, or at least different roots in different trees.
I have a quick question. Just
Yes.
The data set you presented was for one patient.
That's right.
So what do you mean by population? I wasn't clear.
So by population, I mean a relatively homogeneous collection of cells.
Seems like a cell.
So it's a set of cells which are relatively homogeneous compared with the other. So it's part of a tumor.
So do you expect the tree will be different for different cancer?
Yes, and also for different patients. It's really kind of a sort of random process that happens, and there are recurring patterns in terms of, for particular cancer types, which mutations tend to occur in different points, but it's definitely different, in every case.
All right. Thanks a lot.
Thank you.
Next, President Mendel, would you like to go next? Yeah.
Oh, good, thank you.
Yeah, the problem is this, so in order to
Thanks.
yeah, no, actually, oh, I don't know if that works. Try it out. Oh, yeah, that works.
President Mendel is also the president of Cytel, so.
Okay, so thank you very much. So as some of you know, I have a company here called Cytel, and we have a strategic consulting group. And I thought what I'd do is just show you a few of the, three actually, of the consulting projects that our strategic consulting group have been working on. So the first one is what we call streamlining go, no-go decisions. This is a project we did for AstraZeneca. They wanted a method that would sort of automatically work on a visual method to automatically decide which compounds to promote to phase 3 and which compounds to kill. So, so they have for each of these compounds they specify a target value where they think that they can sell the drug if they can beat that target value. For example, a hazard ratio of 0.68 or better, that's a target value. And then they have a lower reference value, which is a real terrible bad value, such that if they don't even meet that threshold, then that's a stop. So they specify a posterior Bayesian probability, where if the posterior probability of beating the target value is at least 10%, then that's a go. And if it's less than 80%, then that for the reference value, then that's a stop. So when they give you numbers like that, you can visually put up a stop light like this, a red zone, which is a stop criterion, a green zone, which is a go criterion, and a yellow zone, which is maybe. So, and on the x-axis is the data. When you see the data, And they can do this for every single compound. They see what's the real hazard ratio that they've observed, and is it in the stop region or in the green region for go or the yellow region. And they also can play around with these numbers, so we can show them, well, what's the probability of being in that yellow region, red region, or green region, so that the idea is to make that yellow region as small as possible, so that they don't have ambiguity, and so they can play around with this visual and come up with a criteria on a standardized scale for all their compounds. Now I'm going to my second consulting projects that we do. We do a lot of these. These are called promising zone designs. What happens is that there's a lot of pressure in a pharmaceutical company to get that drug out quickly. And they want to have as a sample size that's reasonably small, so that they don't have to spend a lot of time or resources. On the other hand, they want to have a good probability of success at the same time. And so they don't want to start with a large sample size. They want to have a strategy of start small and ask for more if necessary. So here's a typical graphic to show that. You have an interim analysis. At that interim analysis, you partition the data into these different zones. There's an efficacy zone where if the data are really good, you can stop right there and claim efficacy. If the data are really poor, you can stop and claim futility. And in between, you just go to the planned end of the study. But if you're in this so-called promising zone, it pays to increase the sample size. And the investors, typically these are small biotech companies in South San Francisco that are looking for investors. And investors don't want to put up the money up front. So they say, well, if you can show that you have promise at the interim analysis, then we put up the rest of the money, and then we'll allow you to go all the way to an increased sample size. And we've done these for several trials. You can see the kind of comments that we get here. Here's a comment from Royalty Pharma, which is a big investor in these types of trials. They say, well, this was a win-win for the sponsor as well as a win-win for us as investors. And then here's a comment from the sponsor of this trial. So that's my second example. My third example, this is a new thing that's a new phenomenon now in the clinical trials world. It's known as platform trials, where you don't want, each company is doing its own trials. Instead of that, why not bring all the companies together on a common platform? And then you would have fewer patients going onto placebo arm and more patients going on the treatment arms. And so these are broken up into two types: umbrella and basket. Umbrella is where you have multiple drugs, and basket is where you have one drug and multiple multiple indications. And so here's an example that we worked on the Stampede trial, multiple treatments, and all from different companies against a common control. Thank you.
We have a couple questions.
The platform trial design is very interesting. And then, so when you consider the common control for the different drugs, do you think that the common control will be comparable for different drug groups?
There may not be. There'll be a common control in the umbrella situation. In the basket situation, there could be a common control, or it may not be. The basket is a little less well developed than the umbrella. It's like you have Keytruda, for example. It's a very big blockbuster developed by Merck, and you can use it for lung cancer. You can use it for ovarian cancer, for colorectal cancer. It's all targeting the same genetic signature, but in different. And you could have a drug, a sort of a generic drug that is just that is currently the standard of care for all of these. but this one you try to get an indication for all of that's what a pharmaceutical company wants, one drug which is an indication for many different tumor types. The trouble is you can't get strong control of type 1 error with this, and so there's some challenges. Yeah.
Are you mainly seeing this sort of trial, Cyrus, in oncology?
These ones are mainly in oncology. The first example of go, no go was for all. The second one, where you're looking at the data and then deciding to increase the sample size, oncology is the main area, but there's a lot of interest now also in cardiovascular trials.
I had a question. Do we have time?
Sure.
Okay. So is there, with the second one, the promising area where you increase the sample size, is there any ethical issue in like letting the investors know it's promising but not favorable?
Yeah.
So you're not stopping the trial. You're not saying we're going to continue without increasing the sample size. So you're giving some information.
That's a key issue of avoiding operational bias. Many of these trials, one way you avoid operational bias is they are double-blind trials. Mm-hmm. So the investigator doesn't know. All he knows is the trial was the sample size was increased. Doesn't know yet is he treating the patient on the control arm or the experimental arm.
Right.
And some investigators might feel actually what we've observed in these types of promising zone trials is that if the word gets out that the sample size was increased, the enrollment increases very rapidly. There's a lot of encouragement. People like to participate in such trials. But double blind is important.
All right. Thanks a lot. All right. Next we have Professor Irizarry.
We'll be entering this.
Convenient to come by the bar.
What? What's going on? You're live.
I can use this to make
I have 5 minutes.
5 minutes, we got you.
Okay, so this is, can I click on, how do I click on something?
I don't.
Oh, like to literally click.
It's okay, I don't have to do it, you know.
Yeah,
No, it's okay, you can just,
can go.
no, it's too much.
No, okay.
You, if I have.
Sorry.
All right, yeah, so I'm gonna, if you want to, I'm Rafa Irizarry. I work at the Dana-Farber Cancer Institute. I'm a professor here too, and I'm here to tell you about a problem that I have. I need help with. So I that there's all these statisticians together to see if I can get some help. And I'm also going to tell you a little bit about what I do especially for students, in case you want to come work in my lab. So there's my webpage if you want to find all the details up there, and I also write sometimes on a blog some statistics. And I also often put updates up on Twitter, and that's the handle. Okay, so what we do in the lab, generally speaking, we work in genomics. Although my interests are in any kind of applied statistics I'm interested in. I did my thesis in musical sound signals. I didn't know anything about biology until I got a job in a biostat department. So if you are interested in any kind of applied statistics problem that you think I might be interested in, I'm also open to anything like that. So I teach a data science class here where we talk about all kinds of different applications of data analysis and statistics. All right, so, but most of my work, the grants that I have, the work that I have to do, relates to genomics, where they have new technologies to measure outcomes of the cell and other physiological outcomes that have never been measured before. So it's a little bit like the invention of a lot of the current technologies. I make an analogy to the invention of the microscope or the improvement of the microscope, where, for example, people discovered microbes. No one saw microbes before the microscope. And the person who discovered microbes was just a guy who was very good at making microscopes get in focus. So that I think is a similar situation to the one we're in now, where you have all these new technologies in genomics that can measure very hard to measure things, and the signal that comes out is very, very noisy, and it could use a lot of help from people who know how to analyze data, how to separate noise from signal, etc. So what we do is take very noisy output from new technologies and make them a little bit better so that biologists can discover stuff. So if you come work with us, you'll be able to sit down on perk, not on the floor, but on a chair. And then another thing we do is that's very specific, very particular about my lab. We try to make everything we work on. We try to work on problems that others want a solution to, and we make our software open source. On the bioconductor project, this is a project we started about 15 years ago now, 17 years ago, wow. And it's quite popular. We get about 27,000 downloads a month, the project as a whole, and it has now about 1,000 packages in it. So that's another very specific thing to our work, is that most of what we do eventually ends up not just as a statistics paper, but also as a piece of software. All right, so now I'm going to ask for help. So one of the projects that I have now is, how much time do I have? Not too much.
1 minute 40 seconds.
Oh, I won't be able to say much. Yeah, so let me try to tell you in 1 minute and 40 seconds. So this is from the data we have, for those that know, is ChIP-seq. It's like ChIP-seq data. We have a YT and a YC, and we also have multiple samples, and we also have it for several locations. All right, so that's what we have. And something that has been done in the past for data like this is you take the log ratio of those two things, and you can explore and fix problems that you have. And here's an example of a plot like that. This is the log ratio of two things that are from genomics, and then on the x-axis is the average. It's the average of the two. Okay, so this is like a scatter plot that's been turned, and this is a control data set. So all the data should be at 0, and there should be no difference. But you see this kind of weird curve, and it's shifted down. The first thing that people try to do is just to shift it up. That's an easy thing to do. But if you shift it up, you still have that curve. So then statisticians did a few things. One of them was to say, why don't you just use the OS, right? You just put the OS right through there, and it fixes it. And that's quite a popular method that will take data like this. These are actually signals. Real signals. You don't want to wash those out. You see the curve, you fix it with OS, and now it's straight. You get rid of a lot of false positives. So that approach works, and I thought I could use it here, but I can't. This is what this data looks, the new data that I have looks like. So you can see. Oh, that's it. All right, well, thank you.
Take 1 more minute, or
1 more minute, okay.
Yeah, that's okay.
If someone could ask them a question like, what are those?
What's on the next slide? You see the curve? It's there. It's there. The curve is there. The problem is that if we fill OS, this is the signal up here. There's a lot of signal in this one. In the previous one, it's just a little bit of the signal is just a small percentage of the points are signal, so you can kind of assume that most of it is something you can assume is 0. We can't do that anymore with this data set. There's so much up there. So we want to be able to fit a curve that fixes that, and then proceed. So here's the problem. These guys are, if you look at the data, it's kind of Poisson. You have a lot of zeros, a lot of ones, a lot of twos, a lot of threes. You kind of see that down here. This is now a scatter plot. So first thing, so these are Poisson, and we can't just fit low X. We kind of have to fit a couple of low S's, one for the signal and one for the noise. So here's a quiz for the students. If this is Poisson with rates alpha t and alpha c, what's the distribution of yt given yt plus yc?
Binomial.
Very good. You're learning statistics in this place. Binomial? Was that a student who said that? Or was it professor? Okay. So is that lt plus c, comma n, equal to n. All right? So that's the first trick we're using, because then that's what now we're plotting this versus this. Right? And you can see that it's kind of close to a half. That's where it would be under the null, that there's no signal. Right? This would be a half. This is a slope of half here. It's not quite there. There's a little bit of a curve. But we can fit something now. We fit something to that, and then we fix it. So now we have, I'm going to call this pi. So now for each sample, we also have a pi at I. J. No, not J. Yes, J. Because it depends on the location. So now the question is, I want to be able to, here's a question I have for you guys. I want to be able to, I have several samples. I want to form some kind of wall test, a t-test, something where it's kind of like a normal variable, using let's say you have five controls, but I have this bias. So that's where I'm stuck, and this is something I'm happy to talk more about, and I'm looking for collaborators that could help me with some ideas of how to do this. Okay, I'm going to stop there.
So maybe if you have an answer, you can find Professor Irizarry after that.
Or an idea.
Or an idea. Okay, so I think, yeah. Lastly, we have Professor Huttenhower.
I want to thank Rafa for covering me, and I'm happy to report that our very sick kitty is getting a little better. So apologies, I had to run out for the phone call. And I have my own timer here, just in case. I'll make it very prominent here. There we go. Alrighty, so our group works on studies of the human microbiome and microbial communities. In the context of the School of Public Health, a lot of what we do is studies of the microbiome in large populations that look a lot like other types of molecular epidemiology. So we have several ways now of generating omics data from the human microbiome in large populations. Much of the time this is sequencing, different variants on high throughput sequencing. Increasingly, some of it's not. We'll look at proteomics or metabolomics of microbial communities associated with the human body and health outcomes. So over the past several years, we've had an opportunity to develop protocols for generating these data, computational bioinformatics methods for generating interesting features from each of these data types. So parts lists of microbes or microbial gene products. We have downstream statistical methods for then associating these features with health outcomes in the originating population. Slowly we're getting enough information to start meta-analyzing these studies and combining results from multiple microbiome population studies of the same phenotypes. And then we do a little bit in our lab and work largely with collaborators on additional validation downstream of mechanisms of how the microbiome influences these host health outcomes. One of the big studies that we've been working on recently that follows this general pattern is the Human Microbiome Project, or the second phase, the HMP2, in which we followed 100 individuals for 1 year each with inflammatory bowel disease. So if you see IBD, this is a GI inflammatory condition that's broken down into two main subtypes: Crohn's disease, which was about half of our patients, ulcerative colitis, which was about a quarter, and then we have about a quarter of the remaining population was controls. We followed everybody for 1 year each. A portion of the population was pediatric, a portion were adults. And the main sample type was, of course, stool samples. You can't have a lunch talk about the microbiome without at least one of these slides. So we have a whole lot of samples in the fridge, in addition to GI biopsies and blood draws, primarily at enrollment. And these really tell us a wide range of molecular features of the microbiome and of the host molecular response to changes in the microbiome during this, and changes in inflammation over the course of a year each. If you take a look at the time courses to get a sense of what the population looked like, within each of these three main phenotypic groups, we have dense longitudinal sampling for a subpopulation. We have sparser longitudinal sampling for the rest of the population, in which we've aligned all of these samples at several time points, such that the NIH mandate was to make multi-omics. Well, we have multi-omics. We have around 300 samples with at least 6 different molecular data types that give us different slices of the microbiome and how the host gut is interacting with it during the disease and treatment. And up to several thousand samples that have been sequenced metagenomically, and intermediate numbers that have some combination of metagenomics, metatranscriptomics, viral profiling, metaproteomics, metabolomics, and fecal calprotectin, which is a host measure of gut inflammation, in addition to these other host-derived data types. So a lot of these data are hot off the press. We have a couple of preliminary results in different slices of early data that we have from the pilot stages of the study, such as a close look at metabolomic interactions in the gut with microbial metagenomic profiles, where we can identify differentially abundant small molecules or small molecule classes. So these are groups of compounds that are mostly the product of microbial biochemistry in the gut. These are enriched or depleted individually during inflammatory bowel disease, Crohn's disease, and ulcerative colitis. But it gets really interesting when we take these individual measurements and start linking them to each other. So in many cases, we can find bugs that are more or less abundant during the disease that also correlate with compounds that are more or less abundant. And sometimes these associations are so strong and above and beyond just what you'd expect from the disease that it suggests potential molecular mechanisms. These could be bugs making the compound or metabolizing the compound, or interacting ecologically with another bug that makes or metabolizes the compound. And in a few cases, such as this example here, Ruminococcus navis, we've been able to do some computational gazing at the metagenomes to understand a little more about why a bug and a compound might be implicated in disease. So for example, for Ruminococcus navis, and that was my 5-minute mark, for Ruminococcus navis, we have a subset of strains that only occur in Crohn's disease patients. They're characterized by about, They're characterized by about 200 genes that make them unique. A few of these genes, we know what they do, and most of them we don't yet know. So there's a nice computational biology problem in now figuring out what all of these disease-specific, microbe-specific genes might be doing. This is a small part of a larger school platform to make microbiome population studies easier. That includes ways to get these samples cheaply, make data from them, and then do the downstream analysis. So if you're interested in facilitating microbiome studies or doing a microbiome study, talk to me or any of the other faces on this particular slide. So with that, I'll wrap again. Thank the folks from the lab who did the work that you saw going by, and our collaborators. Thanks, everyone.
We have time for maybe a couple questions.
It was at 6 minutes. So I technically have 1 minute left. I don't know if I burned it with my phone call. Question?
So have you done microbiome study for cancer?
Only peripherally. We have an ongoing study now in Lynch syndrome as a genetic model of colorectal cancer, where we're doing the analysis right now.
Mm-hmm.
We've done some sort of simple enrichment studies with Wendy actually and Andy Chan, looking at bugs that are enriched during colorectal cancer. But it's difficult to get prospective data to know kind of chicken and egg. So Wendy is looking more closely at that in animal models, and then the Lynch project is where we're looking at that computationally.
Yeah, because I was at NCI yesterday, and it's really impressive to see the signal you have found for non-cancer diseases. But NCI, they have done about thousands of samples, and
Yes.
with microbiome, they did not find much. So I wonder whether you have any insight why it is harder to find signal for microbiome for cancer.
In colorectal cancer, there are some pretty clear links. In other cancers, there's been interest in sort of tissue-resident microbes, and not a lot has turned up there. Where there's been a much closer link is in interactions with cancer treatments. So either through chemotherapy and bugs metabolizing chemotherapeutic agents, or through immunotherapy and bugs mediating modulating the effect that immunotherapy has on immune targeting. Both of those are really hot, active areas, but they're mostly in the gut, not directly at the site of the cancer.
Your data were showing the Crohn's disease versus normal patients, right? So
I was wondering if you had any, or you could collect data where you had different treatments that intervene and show a switch or a change from what it looks like, what the gut looks like on Crohn's disease versus after treatment, if it switches back to normal. That could be a good endpoint for a clinical trial. And I was wondering if there are any thoughts on that.
We have some data on that in the HMP2 because of changes in treatment or responses to treatment over the 1-year time courses. In general, for that and other population studies, medications broadly are of a lot of interest in microbiome studies because unlike genetics, your bugs change. And pharmaceuticals, medications are one of the bigger effects. If you take antibiotics, regardless of what your actual health phenotype is, that has a big effect on the microbiome. So in most of our studies, we have medication covariates. It's not usually, most typical IBD treatments don't have individually huge effects except for antibiotics. One of the other cases that's kind of known in the field is type 2 diabetes. Metformin actually has a big effect on the microbiome, independently of the type 2 diabetes phenotype. So there's a lot of work now looking for other cases like that where missing variation is explained by pharmaceutical exposures.
All right, let's thank all our speakers.